Hello, everyone. I am Alejandro Nato, an assistant professor in Marshall University at Huntington in West Virginia, and I'm here to talk about identification of variants for familial combined hyperlipidemia. Familial combined hyperlipidemia, or FCHL for short, is a complex disease that is characterized by high levels of cholesterol and triglycerides. It presents a relatively heterogeneous etiology, and our goal is to identify single nucleotide variants, or SNVs, that are associated with familial combined hyperlipidemia within regions with evidence of linkage to the disease. Our hypothesis is that variants associated with familial combined hyperlipidemia may affect gene function or lipid metabolism and contribute to its etiology. Here, we report the regions of interest, or ROIs, that were identified through linkage analysis. First, familial combined hyperlipidemia extended families were ascertained by identifying probands with plasma total cholesterol that are at least 95th percentile and triglycerides are at least 90th percentile for age, sex, and race. And the family should have at least 2 participants with familial combined hyperlipidemia and 1 unaffected relative. So our total data set has about 638 individuals in 41 extended families from Charleston Area Medical Center, West Virginia University, and University of Kentucky. We excluded families who only had singletons without DNA or only have 2 to 3 individuals with DNA. And also, after looking at all of these families, we tried to focus on pedigree branches having affected individuals who are closely related. So for example, for this extended pedigree, you will see here that there are 4 affected individuals in this branch, and there are 3 affected individuals in this branch. They're somehow clustering. But for this pedigree branch, there's only a singleton. So what we do is we actually trim that. We remove that and we trim some individuals. So after trimming, we now have a data set that contains 315 individuals in 23 families with family sizes ranging from 4 to 37. And the age range from 2 to 95 years old. And you could see here the total cholesterol levels and as well as the triglyceride levels. Using samples of 180 individuals in these 23 families, we conducted genotyping as well as whole exome sequencing. Using the genotype data, we used the pedigree-based analysis pipeline, or PBAP for short, that I developed to perform file manipulations, validate pedigree relationships, and also to subselect linkage disequilibrium reduced panel of markers. We also use this pipeline to access GLOTA of the Morgan package to sample inheritance vectors, or IVs, which represent the flow of founder alleles in the pedigree. Using these sampled IVs, we perform linkage analysis through PBAP, which accesses GLOTs of the Morgan package to identify regions of interest, or ROIs. The boundaries of these ROIs were determined relative to the maximum LOD score of each ROI, where y equals peak LOD score minus 1/3 of the peak LOD score. Using the LOD scores from these families, we have identified 5 regions of interest with evidence of linkage to familial combined hyperlipidemia on chromosomes 5, 8, 10, and 19. As you can see in the figure, for all 4 chromosomes, these are suggestive hits. 4 of the 5 suggestive hits actually have LOD scores above 2. For regions of interest on chromosomes 5, 10, and 19, specifically at 19q12, which contain 226, 122, and 15 genes respectively, there is no gene found to be associated with FCHL. For the other region on chromosome 19, which is at chromosome 19, P13.2, this ROI contains 221 genes, 12 of which have variants associated with FCHL. And for the other region of interest on chromosome 8, none of the 75 identified genes was found to be associated with FCHL. However, a part of this region has been recently investigated as well, and there were variants associated with FCHL that are intergenic or in introns or actually in transcription factor binding site. Within these regions of interest, we are currently performing family-based gene type imputation in individuals without whole exome sequencing data. So these would be those individuals that don't even have DNA. And we will be able to do this by accessing or using the inheritance vector information. And using known and imputed data, we will perform family-based association analysis to identify variants that are associated with FCHL. And then we will also prioritize the variants using an approach that combines variant information from variant prioritizers, integrators, and annotations that we are currently developing in the lab. Thank you very much.